We'll be discussing the metabolic activities of microbes, especially bacteria, with the aim of trying to figure out how we can best grow them in the lab and also how we can limit their growth outside of the lab. But before we get to that, it's important that we do a quick review of metabolism. And this will be a review. If you want more information on this material, go back and watch the videos from Biology 111 on basic chemistry and enzymatic activity, and also take a look at the cellular respiration lectures from Biology 112. We're going to start today with a very brief overview of basic chemistry and basic organic chemistry. We'll talk about what metabolism actually is, and then we'll look at bioenergetics and the functioning of enzymes. That's going to be followed by a discussion of how organisms, like ourselves, liberate energy from food. And this is broken down into glycolysis, the Krebs cycle or citric acid cycle, and the electron transport chain. And then finally, we'll look at what happens if oxygen is not available. So we'll look at the basic requirements to keep bacteria and other microbes happy. Atoms differ in their affinity for electrons, and we call this electron affinity electronegativity. Now what you're seeing here is a modified periodic table where the y-axis represents the relative electronegativity of different atoms. If we bring two atoms close together that have very different electronegativities, the one with the higher electronegativity will pull electrons away from the atom with the weaker electronegativity. So take a look at nitrogen and oxygen shown in purple here. They have relatively high electronegativities. Compare that to hydrogen and carbon, quite a bit lower. The food that we eat is comprised mainly of hydrogen and carbon. So we have these very complex macromolecules with a carbon backbone and a lot of hydrogen stuck to that, and the occasional oxygen and nitrogen and so on, of course, but mostly carbon and hydrogen. We breathe in oxygen. We combine that with the carbons and hydrogens that we pull off of our food to make carbon dioxide and water. And in the process, electrons are stripped away from the hydrogen and carbon, but not completely, but they're pulled closer towards the oxygen that they end up being bound to. Why is that important? Well, whenever electrons move closer to the nucleus of an atom, energy is liberated. And this set of events is the entire basis of cellular respiration. It's how our cells liberate energy from our food to do work. When a pair of electrons is shared between two atoms, we end up with something called a covalent bond, holding those two atoms together. And we can have two different flavors of covalent bonds. We can have nonpolar covalent bonds or polar covalent bonds. Which one results depends on the relative electronegativities of the participants. So first of all, let's say that we have two atoms that come together that have similar electronegativities. Oxygen, for instance, typically exists in the atmosphere as O2, a diatomic oxygen molecule, two oxygen atoms stuck together. Now, both of those atoms, of course, have identical electronegativities. The electrons are going to be shared equally. In this example here, you're seeing methane, CH4. Now, carbon and hydrogen don't have the exact same electronegativity, but it's fairly close. And that means that the electrons will be shared relatively equally. In a polar covalent bond, we're dealing with two atoms coming together that have quite different electronegativities. Here you're seeing water. So we have an oxygen atom that is attracting two hydrogen atoms. And we're sharing pairs of electrons, but the sharing is uneven. Because oxygen has a higher electronegativity, it has a higher affinity for those shared electrons, and it pulls them closer towards its nucleus. So you can think of electrons, of course, as always being in motion. Well, they're a little bit closer to the oxygen nucleus, and as they're whipping around these atoms, they spend a little more time in proximity to the oxygen nucleus. And what that means is the oxygen, compared to the hydrogens, is slightly negative. We're not talking about a full-on differential of one charge. So we're not talking about oxygen being completely negative compared to the hydrogen, but there is a slight difference. And that's known as a dipole, and that's what that weird little symbol is there. So the oxygen is slightly negative, the hydrogen is slightly positive, and again, that's because the electrons are pulled closer towards the oxygen and they spend more time around the oxygen. So if we were to take an instant snapshot of a molecule of water, we would expect on average that the hydrogens would be slightly positive and the oxygens would be slightly negative. If we take water molecules and throw them together, they are attracted to each other, and that's because, again, the hydrogens with their slightly positive charge are attracted to the oxygens with their slightly negative charge. And this gives water some really unusual properties. It grabs on to molecules that are charged or slightly charged, and it will dissolve them. So this makes water an excellent solvent. The universal solvent, in fact. Also, water will expand when it freezes, which is unusual, although, of course, we're used to water, so we don't recognize that as unusual, but most fluids don't do that. They get denser when they turn into a solid. Also, we have cohesion and adhesion, and the fact that we have hydrogen bonds that need to be broken means that we have to put a lot of energy into water to make it change forms, to make it evaporate. So there's all sorts of very, very unusual properties to water. And these are properties that make life possible, as we talked about in Biology 111. So please, if you need to, go back and review that material. This table shows the bond energies for several common bonds that are found within organic molecules. But what is bond energy? Well, it's the energy that's required to break a bond between two atoms. Or, conversely, it's the amount of energy that's released when that bond forms. So within your body, you are constantly, in every single cell, breaking molecules apart, which requires energy, and then building new molecules, which releases energy. Now, in this table, non-polar covalent bonds are shown in green, and polar covalent bonds are shown in red. When you take in food, you are taking large molecules and you are breaking them down into their monomers. So you're breaking proteins down into amino acids. You're taking carbohydrates and breaking them down into monosaccharides. That requires energy. So the activities of your digestive system require energy. But then those simpler molecules, those monosaccharides and amino acids, are going to be transported through the blood to every single cell in your body, where they'll be broken down further and where energy will finally be extracted. And the reason energy is going to be released is that even though it requires energy input to break those predominantly non-polar covalent bonds in the food molecules, we're going to get a lot more energy back when we form new bonds, when we form these polar covalent bonds in water and in carbon dioxide. So those amino acids and those sugars are going to be broken down bit by bit. The carbons and hydrogens are going to be pulled off. They're going to be stuck to the oxygen that you've breathed in to manufacture water and carbon dioxide. And we're going to get a lot more energy back than we invested. What you're seeing here is the breakdown of glucose through cellular respiration. Now keep in mind that we don't need to use glucose as a food source. There are other options. Also realize, as we'll see, that within your cells, this doesn't happen as a one or two-step process. There are many, many smaller steps in this entire breakdown. So on the left, we have our inputs or precursors, glucose, which is a rather complex molecule, and then diatomic oxygen. On the right, we have our outputs, and they're both quite simple molecules, carbon dioxide and water. So we have an increase in entropy. We're going from a situation of order on the left to a situation of disorder. Remember that when we have an increase in entropy, we have a liberation or release of energy. Energy that perhaps could be used, but more often goes towards random motions of molecules, what we think of as heat. On the left, bonds are being broken. So we have to break the bonds between the carbons and hydrogens and oxygens to free up individual atoms that can then be recombined to create carbon dioxide and water. Underneath glucose there, you're seeing a list of the different bonds that need to be broken. So we have 7 carbon to oxygen bonds. Each one of them, for a mole of glucose, has a bond energy of 78 kilocalories. If we similarly take a look at all the bonds within glucose, we realize that we need an input of 2,182 kilocalories of energy to completely break apart 1 mole of glucose into its constituent atoms. Likewise with oxygen as a diatomic molecule, if we want to break that apart into individual atoms, we need 696 kilocalories of energy. Together that gives us a total of 2,878 kilocalories of energy that are needed to get this reaction started. On the right, we're forming new bonds, and the bonds that we're forming, so carbon to oxygen, oxygen to hydrogen, are polar covalent bonds, and they have higher bond energies. You can see that we have 12 double bonds between carbon and oxygen that will be formed by the breakdown of that glucose. And that's going to liberate a total of 2,244 kilocalories of energy. We're making some water as well. So we're making 12 moles of oxygen to hydrogen bonds, and that's going to liberate 1,320 kilocalories. The total on the right-hand side is 3,564 kilocalories. So we're getting more energy out than we put in. And again, this is such an important process because this is what makes life possible. If we take complex molecules, break them down, and those complex molecules are made up mainly of nonpolar bonds, we take the constituent atoms, we stick them back together, and form much simpler molecules that consist mainly of polar covalent bonds. Well, if we do that, we can get a lot more energy out than we had to put in. Recall that in a metabolic pathway, we start off with a precursor, glucose for instance, and that's going to be processed through a stepwise series of reactions until we have an end product of some sort. We have several intermediates. So we have a precursor, and then we have a reaction that gives us an intermediate, we have another reaction, etc. Each one of those reactions is catalyzed by an enzyme. And usually it's a different enzyme. These metabolic pathways are linked together into a network of interactions that we refer to as metabolism. So for instance, if we look at the breakdown of glucose, glucose would be our precursor. And glucose in this diagram here would be this dot up here. Each one of these dots represents a molecule, and the line connecting 2 dots together is a reaction of some sort. So glucose can be modified and rearranged and broken down through a series of steps along this pathway that makes up carbohydrate metabolism. And those first steps that I've highlighted are part of glycolysis. After that, the intermediate can be passed into a new cycle, that loop that you see at the bottom, that makes up the Krebs cycle or citric acid cycle, and things can be broken down further. But note that any of these intermediates can be taken out and used for other things. So intermediates can be taken out and used to create nucleotides, or used to create lipids, or used to create amino acids. Note also that if we run out of sugar, then amino acids can be shunted into this carbohydrate metabolic pathway and used as an energy source, or lipids can be shunted in, etc. So all of these different pathways are linked together to form a web. Another thing I'd like to remind you of is the difference between catabolic reactions and anabolic reactions. Catabolic reactions occur when complex molecules are broken down into simpler molecules. So the example we talked about, where glucose is broken down and used to create carbon dioxide and water. Those are simpler molecules, so complexity or order is decreased, or the other way to think about that, entropy is increased. And in those sorts of reactions, we get a release of energy. We get the liberation of energy that could potentially be used for other reactions, such as anabolic reactions. Anabolic reactions are the opposite. In anabolic reactions, we take simple molecules and use them to build more complex molecules. So order is increased, or entropy is decreased. So we could use the energy from the breakdown of glucose, for instance, to take amino acids and stick them together to produce proteins. And if it helps, think of anabolic steroids. You take those to build muscle, so you're building something when we're talking about anabolic reactions. In order to shuttle energy from catabolic reactions that release it to anabolic reactions that require it, your cells use ATP, adenosine triphosphate. So we have adenine, a base, attached to ribose, attached to 3 phosphates. Tri indicates that we have 3 phosphates. This is an RNA nucleotide. In fact, it's used in the production of RNA, but it serves many important functions. What's happening here is that this molecule can be used to transfer energy. It contains a lot of potential energy within the bonds between the phosphates. It takes a little bit of energy to break those bonds, but immediately after they break, this phosphate will combine with oxygen, and that releases a lot of energy. Catabolic reactions supply the energy needed to take inorganic phosphate, PI, and stick it to ADP, adenosine diphosphate, to make ATP. In the process, we've created a new bond that will store energy. That stored energy can then be used to power a nearby anabolic reaction. ATP is constantly being used and regenerated. It stores energy for very short periods of time. In fact, a typical human cell goes through about 10 million ATP molecules every single second. So this molecule is recycled between ADP and ATP. The entire AT pool in a typical human cell is recycled or used up in a single minute. If cells require longer term storage, then what they do is they use ATP to build molecules like lipids. Lipids are composed almost entirely of carbon to carbon and carbon to hydrogen bonds, which of course are nonpolar and store a lot of potential energy. Chemical reactions in cells are catalyzed by enzymes. A catalyst is something that speeds up the rate of a reaction. Enzymes are not used up in the reaction. They're not actually reactants. Instead, they grab onto the reactants and they manipulate them in some way. In this example here, you're seeing the breakdown of sucrose. Sucrose is a disaccharide. It's made up of two monosaccharides, glucose and fructose. Now you could take some sucrose and you could throw it into water and it would break down eventually. What's going to happen is occasionally a water molecule will collide with that glycosidic linkage that's holding glucose and fructose together, and it will break them apart. But that's fairly unlikely to occur. The water molecule has to come in at just the right angle. It needs to hit that glycosidic linkage. But if you throw in a pinch of sucrase, this is an enzyme that breaks down sucrose. What it will do is it will grab onto sucrose, it will stress it, it actually bends the molecule and exposes that linkage to the water. Now a water molecule is far more likely to hit that bond, and it actually doesn't need as much kinetic energy to hit that bond and break it. For any kind of reaction, there are at least two steps. So if you are burning something, let's say we're in the lab and you need to light a Bunsen burner. You turn on the gas, you get your striker, you make a spark. Now what does the energy in that spark do? Well, what it does is it allows a few of those gas molecules to be broken down. So the energy of that spark went into breaking down gas molecules into individual atoms that can be recombined with oxygen. That second step, bonds forming with oxygen, gives off energy. It gives off the energy that creates the light and heat of the flame. But you had to put in a bit of energy first. That energy that you put in is known as the activation energy. And you're seeing a nice little metaphor here. So we've got a big hill and we're going to push a boulder down it. But we have to push up this little rise to get to that hill. We have to interject a little bit more potential energy before that boulder can roll down the hill. And that's exactly what happens with a catalyzed reaction, or any kind of reaction. We have to put in a bit more potential energy before that reaction can occur. Enzymes, by lining things up just so and making the conditions perfect, decrease the amount of energy that we need to invest. They decrease the amount of activation energy. The reactions that need to occur within cells would occur far too slowly, if at all, if it wasn't for the presence of enzymes. Lysozyme, for instance, is an enzyme that you secrete into your tears, onto the surface of your skin, into your digestive system, etc. This is an enzyme that will assist in the degradation of peptidoglycan. So peptidoglycan, as we know, makes up a major component of the cell walls of bacteria. And by secreting this enzyme, you catalyze the breakdown of peptidoglycan and cause a lot of problems for bacteria. You limit the growth of bacteria on and in your body. Penicillin, as we'll talk about later, is a compound that inhibits an enzyme that bacteria use to make peptidoglycan. So this is a drug that has a similar effect. It doesn't break down peptidoglycan directly, but it's going to interfere with the production of peptidoglycan. Now bacteria fight back. They have an enzyme known as penicillinase, which actually attacks penicillin and helps in the breakdown of penicillin. Believe it or not, there is an international committee that meets occasionally and discusses how enzymes should be named. All enzymes end in the suffix ase, and the preceding part tells us a bit about what the enzyme does or what the substrate of the enzyme is. So for instance, DNase digests or breaks down DNA. Proteinase, also known as protease, breaks down protein. ATP synthase synthesizes ATP. Integrase is an enzyme we talked about in our last topic. It's used by HIV to integrate its genome into your DNA. Reverse transcriptase is used for reverse transcription by HIV. One of my personal favorites is helicase. Now it's got another name, unwindase. It unwinds DNA before it's split by RNA or DNA polymerase. As mentioned, enzymes catalyze reactions that normally would not occur or would occur far too slowly to be of use. And they do this by lowering the amount of energy needed for the reaction. They align the reactants properly, or they might actually put physical strain on a reactant and stress it. What you're seeing in this diagram in blue is the enzyme. And within a cleft on the enzyme, we have the substrate. The substrate is the molecule that the enzyme is acting upon. The site where the substrate binds is known as the active site, and it has a very particular shape. It only allows the substrate to bind, although occasionally it might make a mistake and allow a similarly shaped molecule to bind, as we'll talk about. What you're seeing here, though, is DNA polymerase, and we have a cleft at the top that allows the binding of a template strand of DNA. So this is a molecule that will divide DNA into two strands, read one of the strands, and use that as a template to make a complementary strand during DNA replication. Recall that a polymer is a molecule that consists of many monomers. Poly means many, mono means one. So for instance, proteins are polymers that consist of many monomers of amino acids stuck together. Nucleic acids are polymers that consist of many monomers, which are the actual nucleotides that are all strung together. Carbohydrates like starch are polymers that consist of monosaccharides, etc. Many of the enzymes that we're going to be talking about in this course are enzymes that assist with the linkage of monomers to form a polymer, or the reverse, where we're taking apart polymers into monomers. What you're seeing here is dehydration synthesis. When you see D at the beginning of a word, it usually refers to the removal of something. Hydro means water. Synthesis, of course, means building something. So we're building a polymer by removing water. So you can see here we have a short pre-existing polymer. It's got a hydrogen exposed at one end. We've got a monomer that's coming in. Could be an amino acid, could be a monosaccharide, whatever, but it has a hydroxyl group, an OH group exposed. That hydroxyl and hydrogen will combine to form a water that's being pulled away. That's the dehydration part. And we're forming a longer polymer. The opposite of this is hydrolysis. Again, hydro means water. Lysis refers to splitting or breaking. So we're taking that water, we're pulling off a hydrogen, we're attaching that to the pre-existing polymer, and the hydroxyl, the HO that remains, is attached to the monomer so that it can be removed. Enzymes can assist in joining molecules together or pulling them apart. In this example here, we've got an enzyme that's floating around, bumping into things, and if it bumps into these two particular substrates, depicted by yellow and red, and if they bond into the active site, they will be held in the exact position they need to be in to join together through dehydration synthesis. Now, there's going to be a whole lot of other stuff moving around in the cell. Non-substrates don't have the right shape. They will not bind to the active site. But once again, if we have the two correct substrates coming in and binding to that active site, the enzyme will actively align them in the correct position so that they can form an appropriate bond. The shape and also the charge distribution of an active site will determine what substrate can bind to a particular enzyme. Here you're seeing sucrase again, and sucrase will bind to sucrose. Sucrose consists of two monomers, glucose and fructose. And what sucrase does is it strains the bond, the glycosidic linkage, that links the glucose and fructose together. It exposes that bond to water. It makes this breakdown reaction far more likely to occur. So enzymes can break things down, they can build things up. They can also simply rearrange atoms within a molecule without changing their chemical formula. And enzymes can also transfer atoms, or sometimes just individual electrons, from one substrate to another. And that's what you're seeing here. So we have a substrate known as PEP, and you notice that it has a phosphate attached to it, and that phosphate is being pulled off and attached to ADP to make ATP. And the energy that was contained within that bond is being transferred as well. Of course, we're going to lose a little bit because no energy transfer is 100% efficient. Enzymes that transfer phosphates from one molecule to another are known as kinases. And these are really important enzymes, as you might imagine, in metabolism and also in regulating the activities within a cell. Enzymes are very picky when it comes to the substrate that they can act upon. The substrate has to have the right shape and the right charge distribution in order to fit into the active site. But enzymes are also very picky when it comes to environmental conditions. For any chemical reaction, whether or not it's catalyzed, temperature plays a big role in the rate of the reaction. Imagine that you take a beaker of water and put it on a hot plate and heat it up. You put a thermometer into that beaker of water to measure the temperature. What are you actually measuring? Well, you're measuring the frequency and energy of collisions. So you're measuring the average kinetic energy of the water molecules. If the water is very hot, water molecules will collide with the thermometer more often, and they'll collide at greater speed. They'll have more kinetic energy, and more energy will be transferred to the thermometer. So if we have a solution that's warm, molecules are moving more quickly, they're hitting each other more often, they're hitting each other with more energy, and all of that leads to a greater probability of a reaction occurring. Now, if we have an enzyme catalyzed reaction, and it's happening in a test tube, let's say, and we heat up that test tube, the rate of the reaction will increase up to a certain point. So you can see in the first graph here, when we get up to 40 degrees, all of a sudden the activity drops off. And that's because the enzyme is being denatured. Above 40 degrees, the enzyme is going to lose its shape. And that includes the active site. The active site is going to be the wrong shape as the enzyme denatures. And the reaction is going to dwindle down because we don't have that catalyst anymore. Now, it depends on the organism, but this is the general shape of a graph that we would expect for a mammal like ourselves. pH has an impact as well. So the majority of enzymes prefer a pH that's near neutral. But there are some enzymes that we have in our bodies that like pH to be quite a bit higher or quite a bit lower. Pepsin, for instance, is active in the stomach, which is incredibly acidic. But again, the pH is going to affect the shape of the enzyme, which is going to affect its functioning. Finally, we have substrate concentration. Imagine we have a reaction that's occurring in a test tube and it's catalyzed by an enzyme. We can add more substrate, we get a greater rate of breakdown because we have more reactants that can react. However, we'll get to a point where the substrate overwhelms the enzyme there's simply not enough enzyme to make that reaction occur faster. So we'll get this plateau, we'll get this leveling off that occurs. If we wanted to increase the rate of reaction further, we'd have to add more enzyme. And then we would get this new plateau, this higher plateau that would form. There are other factors that influence the activity of enzymes, and generally it's because it changes the shape of the active site. The salt concentration has an effect. The concentration of the product has an effect. If there's lots of product, a reaction will slow down, or it might even run in the opposite direction in some cases. The organ where this is occurring will have a big influence, and that's because the pH may be different in different organs or in different organelles, and also there may be other factors, other buffers and so on, that are only present in certain organs. When a protein denatures, it changes shape, so its folding changes. And it may be that the folding changes a little bit, or the protein may completely unravel. Heat will cause proteins to unravel. And this is what happens when you cook an egg. So the egg white starts out clear and rather fluid, but of course it becomes dense, firm, and opaque. And what's happened is the albumin that makes up the egg white has denatured. It has changed shape. It's not possible to renature that albumin, although in many cases, for many proteins, they can be renatured if environmental conditions return to what they were. We can use heat to denature proteins, but we can also change pH. And if you enjoy poached eggs, the best way to poach an egg is to use boiling water with some vinegar in it. The vinegar will reduce the pH, and that will help the albumin denature more quickly. And you get nice, compact, really pretty looking poached eggs instead of those weird, wispy looking things. Heavy metals and chemicals will also bind to proteins and change their folding. So for instance, a chemical might bind to charges on the protein and prevent those charged side groups or functional groups from interacting with oppositely charged functional groups. And the folding of the protein may break down as a result, or those chemicals or heavy metals may physically block the active site. And this is how a lot of the cleaning products that you have in your home function. They denature proteins, and by doing so, kill or inhibit the activity of bacteria. A substance that binds to the active site of an enzyme and blocks the substrate from binding is known as a competitive inhibitor. It's competing with the substrate for access to the active site. So it could be that this inhibitor has the same shape as the normal substrate, or at least it has a similar charge distribution and can bind to at least part of that active site. Now, generally, this binding is reversible. In fact, the competitive inhibitor might only bind for a fraction of a millisecond, but during that fraction of a millisecond, substrate cannot bind. So you can imagine that if the substrate is fairly rare in the cell and we overwhelm the cell with a lot of competitive inhibitor, even if that competitive inhibitor is spat out after a fraction of a millisecond, it's going to overwhelm the enzyme. The enzyme is going to waste all of its time with that competitive inhibitor that it can't do anything with, and it will never have access to the substrate. So we can use this inhibitor to shut down a reaction. We also have non-competitive inhibition, and this is where an inhibitor molecule binds somewhere other than the active site. We refer to this site as an allosteric site. Allosteric means somewhere else. So the inhibitor is not binding to the active site, it's binding somewhere else on the enzyme, and by doing so, it changes the shape. It changes the folding of the enzyme. That changes the shape of the active site, and the substrate can no longer bind. This is the means by which a lot of cells will regulate their enzymatic activity. So for instance, let's say we have an enzyme that's involved in manufacturing the amino acid tryptophan. Tryptophan is the product of this chemical reaction, and it also acts as a non-competitive inhibitor. It will bind to the allosteric site of that same enzyme and shut down the reaction once the cell has enough of that amino acid. So once there's plenty of tryptophan in the cell, it shuts down the reaction, which is pretty clever. There are a lot of drugs and chemicals that we might use to inhibit the activities of microbes that work in this way. They work as competitive inhibitors or non-competitive inhibitors. Let's take a look at a few examples of competitive inhibition. The most common neurotransmitter in your body is acetylcholine. It's released by an axon terminal into the synaptic cleft. It crosses the cleft and binds to a receptor on the postsynaptic cell. That causes the postsynaptic cell to fire off an action potential, or if it's a muscle cell, it causes the muscle cell to contract. Now, so long as that acetylcholine remains attached to receptors, it's going to cause that cell, that postsynaptic cell, to continue firing. So it has to be removed, and it's removed by acetylcholine esterase. It's broken into two pieces, and then the axon terminal is going to recycle and repackage that acetylcholine. Sarin is a very powerful, very dangerous nerve gas. What it does is it binds to the active site of acetylcholine esterase, and it blocks the active site so that it cannot break down acetylcholine. Now what happens is that the postsynaptic cell continues to fire. So you lose control of your muscles. You can't relax them. They're constantly in kind of a tetanus situation. What you're seeing in the figure there is the unfortunate aftermath of a terrorist attack in Tokyo back in 1995. These people are unable to breathe because they can no longer control firing of postsynaptic cells. Prostaglandins are signaling molecules that are released at the site of a wound or infection. They cause increased blood flow to an area, and that brings about swelling and pressure, and it also causes pain receptors to fire off a signal back to your brain. Acetylsalicylic acid, or aspirin, competes for the active site of one of the enzymes responsible for making prostaglandins. And by doing that, it alleviates swelling and the pain that the swelling causes. Penicillin binds to the active site of an enzyme that's used to build peptidoglycan in bacteria cells. If we expose bacteria to penicillin, the cell wall is weakened. The bacteria can't maintain that cell wall. Bacteria cells tend to be under quite a lot of pressure internally. The fluid is under pressure, but the cell wall pushes back. If the cell wall is weakened, the bacteria will rupture and be killed. Let's take a look at one final example. Let's look at the competitive inhibition of alcohol dehydrogenase. This is an enzyme in your body that breaks down ethanol. So if you have an alcoholic drink, alcohol dehydrogenase is going to convert the ethanol to acetaldehyde. Acetaldehyde is somewhat dangerous, and it is broken down quite quickly by a number of other enzymes and through a number of different steps, ultimately to carbon dioxide and water. But what if you were to drink methanol instead of ethanol? Well, alcohol dehydrogenase can also grab onto methanol and break it down. It's not as fast a reaction. Alcohol dehydrogenase doesn't have as high an affinity for methanol as it does for ethanol, but if it breaks down methanol, it converts it into formaldehyde. And as you might imagine, formaldehyde really isn't good for you. It's especially harmful to the rods and cones of the eye, and drinking methanol can cause you to go blind. Ethylene glycol can also be broken down by alcohol dehydrogenase into a number of rather dangerous compounds that can damage your liver and other organs. Ethylene glycol is found in antifreeze, and it gives antifreeze a somewhat sweet taste. which is why, unfortunately, it's not uncommon for children to drink antifreeze or for pets to drink antifreeze. So be very careful where you store your antifreeze and how you clean it up if you spill it. Anyway, let's say that someone comes into the hospital and they've had methanol or they've had ethylene glycol. Well, how can you treat that? Well, what you want to do is you want to shut down the activity of alcohol dehydrogenase. Methanol and ethylene glycol are rather small molecules and they will ultimately be removed from the blood by the kidneys. But we want to make sure they're not converted into something more dangerous before that happens. So you can use competitive inhibition to decrease the activity of alcohol dehydrogenase. If someone comes in and they're drunk on methanol, you could give them a whole lot of ethanol. And the ethanol will compete for the active site of alcohol dehydrogenase. And that means alcohol dehydrogenase will spend a lot less time breaking down methanol. So hopefully the methanol can be removed from the body before it's converted into formaldehyde. Now, getting someone really drunk on ethanol that's already drunk on methanol might not be the best way to do this. There are some other drugs that can also competitively inhibit alcohol dehydrogenase. We're going to end off our discussion of microbial metabolism with a quick review of cellular respiration. Most of you have just seen this in Biology 112. If you haven't, you might want to take a look at that video online because it goes over this in a bit more detail. Cellular respiration is the set of reactions that allow cells to extract energy from food items. The very first step in this process occurs in the cytosol, and it's called glycolysis. This is a process that evolved in very early cells, and all cells today share that process. With one exception, chlamydia doesn't bother. As we've talked about, chlamydia has gotten rather lazy as an obligate endoparasite, and it just steals its ATP from the cells that it parasitizes. But in all other cells, we have this first stage of cellular respiration occurring within the cytosol that produces a small amount of ATP. To get more ATP out of our food requires a process known as oxidative phosphorylation, and that requires oxygen. There are anaerobic cells that do not undergo oxidative phosphorylation. They rely entirely on glycolysis. So these are cells that live in an oxygen-free environment. And then we have aerobic cells that live in an oxygen-rich environment and require oxygen. The oxygen in our cells is going to be utilized by the mitochondria. Mitochondria, remember, evolved from aerobic bacteria. Within the mitochondria, we have something called an electron transport chain. This is a chain of proteins embedded within the inner membrane of the mitochondrion. We have electron carriers. These are molecules that transport energized electrons that pass down that electron transport chain. And as they do that, the energy is extracted from those energized electrons, used to pump hydrogen protons into a space within the mitochondria, as we'll discuss. And it sets up pretty much a battery. It sets up a situation where we can produce, using a charge differential, a lot of ATP. What you're seeing here are mitochondria within the muscle cells of a rat. The first step, glycolysis, occurs in the cytosol. Glycolysis literally means breaking sugar. Glyco means sugar, lysis means to split or to break. Glucose is going to be broken into 2 3-carbon molecules of pyruvate. Now, once again, we don't have to have glucose in our diet. Carbohydrates are non-essential, and bacteria don't absolutely have to have glucose either. So if glucose is not present, your cells can build their own glucose from amino acids and other precursors, or they can take amino acids and also nucleotides and lipids and break them apart and take those simpler molecules and insert them as intermediates in glycolysis or intermediates of the citric acid cycle. So this is rather flexible, this system, but traditionally we just start at the beginning with glucose. Okay, that pyruvate is going to be either broken down through fermentation, as we'll see if there's no oxygen present, or two of the carbons of pyruvate will be pulled off to form an acetyl group, and those will be attached to a large molecule known as coenzyme A. That acetyl CoA is transported into the mitochondria. The acetyl is pulled off and stuck into a cycle known as the citric acid cycle or Krebs cycle. Now, when that first carbon was pulled off of pyruvate, it was stuck to oxygen to make carbon dioxide. The rest of the carbons, the two carbons in that acetyl group, will be pulled off of intermediates. As they pass through the citric acid cycle, they will also be combined with oxygen to produce more carbon dioxide. This is where the carbon dioxide that you breathe out comes from. The mitochondrion is a rather special structure. As we've talked about before, it's thought to be essentially an enslaved bacterium. There's two membranes. The outer membrane resembles other membranes we have in our cells, but the inner membrane resembles a bacterial membrane. That inner membrane has a lot of protein in it. In fact, it's about 75% protein. Those proteins are parts of the electron transport chain. Some of them act as pumps, and then we also have ATP synthase embedded within that inner membrane, and it's ultimately going to produce the majority of ATP that we use in our cells. In the very center, we have an area known as the matrix. Lots of stuff going on there too. We have lots of proteins there, lots of enzymes that are involved in the citric acid cycle. This figure provides a nice overview of what happens in the majority of eukaryotic cells. Remember that there are a few eukaryotic cells that don't have mitochondria, Parabalsula and Diplomonadida, but those are odd exceptions. There are also some eukaryotic cells that have mitochondria but quite often choose not to use them. Yeast, for instance, tends to favor fermentation and tends to ignore its mitochondria. Okay, so the first step, once again, is glycolysis. That occurs in the cytosol. And what's happening here is something known as substrate level phosphorylation. This is where phosphates are taken off of a substrate and stuck directly to ADP to make ATP. We have a small amount of ATP that's being generated through glycolysis. Electrons are being pulled off of food items in glycolysis as well and stuck to an electron carrier known as NAD+. So we have two electrons and one proton being stuck to NAD positive to make NADH, and then NADH will enter into the mitochondrion carrying those electrons with it. The pyruvate that results from glycolysis is going to be broken into a free carbon and also an acetyl group, and that acetyl group is going to be transported into the mitochondrion where it will be broken down further in the citric acid cycle. During this breakdown, energized electrons will be pulled off and attached to two electron carriers. This will generate NADH and FADH2. Those molecules carry energized electrons to the electron transport chain, along with the NADH that was produced during glycolysis. Oxidative phosphorylation occurs within the mitochondria by the participation of the electron transport chain. So the electron transport chain is going to set up a gradient of hydrogen protons. Basically, hydrogen protons are going to be pumped into the intermembranous space, that space between the two membranes. That gradient is going to be utilized through chemiosmosis to generate lots of ATP. And this is where 90% of the ATP that we manufacture in our cells comes from. We have a small amount that's also contributed by the citric acid cycle directly. The gray spheres in this diagram represent carbon atoms that were taken in as food. So glycolysis, once again, is going to start with glucose, break it into two pyruvate molecules. Each pyruvate contains 3 carbons, and a lot of other atoms, of course, but we're just focused on the fate of those carbons. Pyruvate is going to be broken into an acetyl, which contains 2 carbons, and a free carbon that's stuck to oxygen to produce our first carbon dioxide. In that process, we also generate the electron carrier NADH and some free hydrogen protons. The acetyl group is going to be stuck to coenzyme A and transported into the matrix of the mitochondria. The matrix is where the citric acid cycle occurs. That acetyl group is going to be broken down further to generate more carbon dioxide. Also, energized electrons will be pulled off, and they're used to generate NADH and also another electron carrier known as FADH2. These electron carriers are going to transport these energized electrons to the electron transport chain. The 2 carbons contained within that acetyl group are going to be pulled off and stuck to an intermediate of the citric acid cycle known as oxaloacetate. Through 2 rotations of this cycle, those carbons will be pulled off to make carbon dioxide, and electrons will be pulled off as well and transferred to electron carriers. After that's complete, we will have generated 3 molecules of NADH, 1 molecule of FADH2, 2 molecules of CO2, and 1 molecule of ATP. Now you're probably wondering why the numbers that I've given are doubled. That's because we get 3 NADH, for instance, from 1 pyruvate. But remember, when we're looking at numbers for cellular respiration, we always refer back to 1 molecule of glucose. So we started with that 1 molecule of glucose. That gave us 2 pyruvates. We get 3 NADH from 1 pyruvate, we double it so that we're always seeing this in terms of that original input of 1 glucose molecule. The energized electrons that were pulled off of food molecules and stuck to electron carriers will now be donated to the electron transport chain. Remember that the electron transport chain consists of proteins embedded within the inner membrane. Those energized electrons will bounce from protein to protein, giving up energy as they do so. That energy is used to pump hydrogen protons from the matrix to the intermembranous space. And this sets up a very strong gradient. It's an osmotic gradient, but it's also a charge gradient. So we have lots of hydrogen protons that are being sequestered between those 2 membranes, and they're repelling each other. They will move back into the matrix through chemiosmosis. However, there's only one place where this can occur. So those hydrogen protons can't simply sneak through the membrane itself. They have to go through a channel, and that channel is within a large complex of proteins, making up a structure known as ATP synthase. ATP synthase is essentially an electric motor. Remember that bacteria invented the electric motor. They use electric motors to pump things. They use them to spin their flagella, etc. And we've been very fortunate in that we inherited these electric motors in our mitochondria. So this very strong gradient of charge and concentration is going to force hydrogen protons into this electric motor. And they're going to fit into slots within the rotor. As a new hydrogen proton comes in, it's going to repel the one that's next to it, the one that's already slotted. And that's because like charges repel, and that causes rotation of the rotor. That mechanical energy is going to be transferred down to this internal catalytic knob, which is kind of an unusual name for this. It's also known as the lollipop head, which isn't much better, but this structure is going to spin, and as it spins, it grabs onto ADP and inorganic phosphate and slams them together to make ATP. So we have this gradient that's being used to create mechanical energy that's being used to create chemical energy in the form of ATP. I can't stress enough how remarkable this little motor is. People that are interested in nanotechnology have been studying this intently for quite a while because it is the smallest motor that we're aware of, and it would be pretty cool if we could build something similar. What's also really neat is that this thing can run in reverse. Let's imagine a situation where a cell has a whole lot of ATP and it doesn't really need it at the moment, and there's a whole lot of ATP in the matrix. Well, the ATP can be used to rotate this in the opposite direction. And now ATP is going to be used to power this as a pump, and it will pump hydrogen protons out into the intermembranous space. So if the mitochondria has lots of extra ATP sitting around, it can use that ATP to store hydrogen protons for a rainy day. Pretty neat stuff. And if you want to see a really cool animation of this, please check out my other video where I talk about ATP synthase in a bit more detail. An oxidizer is a molecule or atom that pulls electrons away from something else. It gets its name from oxygen because, of course, oxygen is a very good oxidizer. It has a high electronegativity, and that means it has a high affinity for electrons. It also means that oxygen is potentially rather harmful to a cell. If it's wandering around in the cell and pulling electrons off of enzymes and other molecules, it could do quite a bit of damage. So the mitochondria not only allow us to oxidize our food and in the process get more ATP, the mitochondria, by gobbling up oxygen, actually help protect the cell from damage. What is the oxygen used for in us? Well, it's used in the citric acid cycle. So carbons have to be pulled off in this cycle in order for it to work, and they're stuck to oxygen to make carbon dioxide. This also occurs when we're converting pyruvate into an acetyl group. But if we look at the electron transport chain, notice that the electrons that were pulled off of electron carriers and bounce along the proteins of the electron transport chain are ultimately going to be stuck to 2 hydrogen protons and an oxygen to make water. So we say that oxygen is the terminal electron acceptor. And I'm talking about this equation right here. So if we didn't have oxygen present, we wouldn't be able to get rid of those electrons, and we have to get rid of them for this process to continue. Looking at the numbers, it's obvious that the vast majority of ATP comes from the electron transport chain. It's a product of oxidative phosphorylation. And you might be wondering, why are there cells that are anaerobic? Why is that a thing? If a cell is anaerobic, it doesn't have an electron transport chain, it can't undergo the Krebs cycle or citric acid cycle. It's restricted to glycolysis to generate ATP. And we only get 2 ATP per molecule of glucose. Very, very inefficient. Well, the secret is that glycolysis is much faster than the electron transport chain. In fact, it's 100 times faster. So in the time that an aerobic cell generates 38 ATP from 1 glucose molecule, the cell that's undergoing only glycolysis can generate 200 ATP. And if there's lots of food available, efficiency might not matter. We have muscle cells that bring about very quick, very powerful contractions using only glycolysis. Now, cells that are anaerobic are only undergoing glycolysis. They have no need for oxygen, and in fact, oxygen is poisonous to them. And that, once again, is because oxygen is a highly electronegative atom. It oxidizes other parts of the cell. The food we eat consists of molecules with a carbon backbone. I'm afraid if you're eating something that doesn't have carbon in it, you're not getting anything out of it. We need these molecules as an energy source, but also we need that carbon to build our own macromolecules. Carbon can come from a number of different places. If we look at photosynthetic bacteria, cyanobacteria, or plants, they take carbon out of the air. They take carbon dioxide out of the air, and then they fix that carbon, which means they attach it to other organic molecules, and then they break down those molecules that they made using the same process we do, cellular respiration. So plants and algae and cyanobacteria make their own food using carbon dioxide that's dissolved in water or in the air. But there are other things that can be consumed by some microbes. Oil and petroleum, PCBs, toluene, which is a very, very dangerous chemical to us. It's quite toxic. But there are microbes out there that can consume these different carbon sources. So remember that during glycolysis, we make a small amount of NADH. We're taking energized electrons off of food, sticking them to NAD positive, and then the NADH is going to transfer those electrons to the electron transport chain, and that's going to regenerate our NAD positive. But what if we're talking about an anaerobic cell? Well, this still has to happen. Electrons still have to be pulled off of those intermediates and stuck to NAD positive, but now the electrons have nowhere to go. There is no electron transport chain. So those electrons have to be pulled off to recycle that NAD positive. And what happens is those electrons are stuck to pyruvate. Pyruvate's not going to be used any further either. There's no citric acid cycle for the pyruvate to pass into. So the electrons are pulled off of NADH and they're stuck to pyruvate. Pyruvate is rearranged to form other products, ethanol, for instance, or lactate. This is a process known as fermentation. And the whole point of fermentation is to reconstruct NAD+ so that glycolysis can continue. In alcohol fermentation, we have the production of ethanol, although other alcohols can be produced in some organisms. And we also have a second byproduct, which is carbon dioxide. Carbon dioxide produces the bubbles in champagne and beer, and it also causes bread to rise. And of course, ethanol provides us with our alcohol content. There are a number of different possible fermentation end products. It depends on the species of organism that we're talking about. In all of these, we start with glucose, and that's converted into pyruvic acid. Electrons are taken off of NADH, and remember the reason for that is to regenerate NAD positive. The electrons are stuck to pyruvic acid, but then there's a number of different things that compound can be converted into. We have Saccharomyces here in the middle. Remember, that's a eukaryotic organism. It's a type of fungus. It does have mitochondria, but it quite often doesn't use them. And that's because glycolysis is faster. It can generate plenty of ATP just using glycolysis. But once again, it has to regenerate that NAD+, so that means that it has to undergo fermentation. And the end product is carbon dioxide and ethanol. We have bacteria that will produce lactic acid. Lactic acid causes milk to curdle, and we can use that to make cheese and to make yogurt. It's also bacteria that's found on your body, as we'll discuss, and it can help maintain a low pH in regions of the body, and that could be quite helpful. We have bacteria that will also live in cheese and generate carbon dioxide, which gives rise to bubbles. That creates the holes in Swiss cheese. We have some bacteria that produce acetone and isopropanol as a byproduct of fermentation. And they're actually used in industrial situations to generate these compounds. And then, of course, there's other bacteria that produce acetic acid, which is vinegar. Aerobic refers to the presence of oxygen. An aerobic environment is an environment where oxygen is present. An anaerobic environment is one where oxygen is not present. Aerobic organisms require oxygen, and they use oxygen as their terminal electron acceptor for all of those electrons that were transported by electron carriers. Again, for anaerobic organisms, they still need to get rid of a small number of electrons that were stuck to NADH, and they do that by sticking it to pyruvate. An obligate aerobe is an organism that requires oxygen for survival. We, of course, are obligate aerobes. A facultative aerobe is one that can use oxygen if it's present, but it can use alternate pathways. So yeast would be an example of this. Anaerobic respiration involves just glycolysis, and there needs to be other electron acceptors. That might be pyruvate, or in some cases, it can be something else, but we won't get into those examples. Obligate anaerobes are poisoned by oxygen. If oxygen is present, it will kill their cells. Now, I realize this topic was entirely review, but it is important that you refresh your memory when it comes to these ideas before we go forward and look at new stuff. Metabolism consists of two parts. Catabolism is the breakdown of large ordered molecules to liberate energy. And anabolism is the opposite. So we're taking energy that was freed up by catabolism and we're using it to build stuff. Nearly everything that happens in a cell has to be catalyzed by an enzyme. The reactions of life don't occur on their own typically, or if they do, they occur far too slowly to be useful. Enzymes have an active site, and the active site binds to a substrate. A number of the antimicrobials and drugs that we'll talk about in this course target enzymes. They inhibit them, either competitively or non-competitively. ATP, adenosine triphosphate, should be your favorite molecule because it is the energy currency of the cell. It's not used for long-term storage of energy, but it's used to shuttle energy between catabolic and anabolic reactions. Microbes have very diverse nutritional requirements, as we'll see. Many microbes can do some pretty amazing things. They can use sources of energy in the environment that we do not have access to. So for the cellular respiration, it was a pretty quick overview. What do I want you to know? Well, I want you to know the different steps, of course. Glycolysis is going to take glucose and break it into two pyruvic acid or pyruvate molecules. Then the citric acid cycle or the Krebs cycle is going to break down those food molecules further, producing carbon dioxide in the process and freeing up energized electrons that can be stuck to electron carriers. The electrons are going to be bounced along the electron transport chain, and that is going to be used to set up a gradient of hydrogen protons. The hydrogen protons, which build up in the intermembranous space, will flow back into the matrix through ATP synthase, and ATP synthase will generate the vast majority of the ATP that we get out of a glucose molecule. But again, remember that there are many cells that do not require an electron transport chain. They are not as efficient. They rely on glycolysis alone. If a cell is relying on glycolysis alone, it still has to get rid of electrons that were carried by NADH. Those electrons have to be stuck to something. Quite often that is pyruvate, and then pyruvate is rearranged to form something else, and that's a process known as fermentation. I want you to know the inputs and outputs of each part of this process. And here's a nice little summary of that. You should know how many electron carriers are produced by each process, and you should also know how much ATP is generated. And finally, here's our terminology. And even more finally, here are a few study questions.